Thank you very much for the kind introduction, and I also would like to take a couple of minutes to thank the organization for organizing such a wonderful conference. At the beginning, I was telling our CEO, Paul Fultel, which some of you might know, how long would my list of competitors be after this conference, to which he said, we are just all friends here. And I really think that nicely showcases the feeling that this conference had, at least for me. So with that being said, I'd like to dive into modeling of ischemic stroke in neurovascular unit on a chip. So to begin, what is the neurovascular unit exactly? To form the BBB, the endothelial cells are by far the most important component. So they are really essential in forming the blood-brain barrier and to make sure that components that are not supposed to go in are not going in, and the other way around as well. Now, there's a lot of other cell types that are also involved in forming that blood-brain barrier, such as the basement membrane, the astrocytes, parasites, neurons, microglia, and oligodendrocytes. Now, when the BBB get disrupted due to, for example, central nervous system diseases, this goes along with the dysfunction of the neurovascular unit. So this is really something that we aimed to model. And one of the diseases that can cause this kind of disruption is, for example, stroke. Now, why is it exactly important to study this type of disease? Well, stroke is the second leading cause of that globally, and it is the leading cause of adult disability. You can imagine that that comes together with a huge financial burden, but also every fourth person over the age of 25 years might suffer stroke in their lifetime. So you can imagine how tremendous that impact actually is on society. Now, what is ischemic stroke? Ischemic stroke represents 80% of all the stroke cases that we see, and it actually occurs because of an obstruction by a blood clot to the flow inside of the brain. And because of this, there is a limited availability of oxygen, glucose, and essential nutrients to that specific region. And once this has happened, the BBB gets disrupted, you get infiltration of immune cells, and also mitochondrial dysfunction. The neurons get overly excited, and there is cell death presence within this disease. Now, very striking is that even though it has such a big burden, there's only one approved therapeutic agent, and it has an extremely narrow therapeutic window. So it needs to be applied within 4 hours of the stroke from happening. So you can imagine before it is actually diagnosed that this is often something that is too late to actually apply. So because of that, we wanted to create a model in which you can actually improve the therapeutics for this specific disease. And in our case, we used the organoplate from Imedas, which you might have seen in some of the previous talks or on some of our posters. And this platform is based on a standardized 384 well plate format, and it consists out of 40 to 96 chips. Now, for the neurovascular unit, we used our 3-lane 40, so that means it has 3 lanes, as you can see in this representation here, and 40 chips within one of those plates. So here is a schematic representation of one of those chips, with the 3 channels being represented here. And the gray bars that you can see are actually the stars of our system. So these are our phase guides. This is our patented technology, which keeps our whole system together. In between these phase guides, we culture the extracellular matrix, and because of capillary forces, the system is kept together. So all of our models start with culturing that extracellular matrix in the middle channel and then gradually adding complexity to the model. So after the extracellular matrix gel is loaded and solidified in that middle channel, we then added astrocytes and neurons in the bottom channel in a 4:1 ratio. And then we waited until they formed the networks that we also see in the human body. So this takes about a week. And after that, we also added the brain endothelial cells here in the top channel. And I like to also point out here that, of course, the system includes perfusion that starts from step number 3. And our system actually works with a rocker-based system that can fit inside the incubator and causes a bidirectional flow to induce that perfusion and hence also the assemblance of the 3D environment that you can see. So this is a phase contrast picture of our neurovascular unit containing the 3 cell types. The white arrows here actually contain, you can see that it is also a leak-tight barrier. And here you can see the nice networks that are formed, again showing here in the schematic representation. In the top channel we have our tubular structure consisting out of the primary human-derived brain endothelial cells. In the bottom channel we have our astrocytes and neurons, and in the middle we have our extracellular matrix. So this is actually a picture of the complete plate, so containing 40 of these chips and 40 models, which is really nice because you have in one system you can apply your different compounds but also your different controls and nicely envision that. In this picture we can also see that it is quite reproducible, showing that all of the models shown here form equally barriers and look very similar from each other. Now, of course, for creating a good model, you also need to check if you have the correct markers, which is quite detrimental. And so here we checked for the markers of the endothelial tubular structures as well as for the astrocytes and neurons. And here you can see a nice representation of the human-derived brain endothelial cells, also pointing out here the clear lumen that we can see within this model, which is important for forming that specific barrier. And then here in the bottom channel we have our astrocytes and neurons, which are labeled by the S100 beta and the tube 3. And then we also checked for the tight junction formation to make sure that our barriers were tight enough to represent that neurovascular unit that we have present within our brain as well. So besides also looking like an actual neurovascular unit, it also needs to be functional for us to be able to test or to model the specific disease. And what we did here is we actually compared the expression of relevant transporters and compared that with what was present in our 2D HB-mix, just the cells that came from the flask, and to the cells which were present within our chips. And what we can see is that indeed the relevant transporters that are important for this neurovascular unit are indeed also contained within our 3D environment. Here with the PGP assay, we are also checking if the calcium is retained. So if this would be disrupted, the fluorescent would actually leak out of this. But we can see that both the transporters are working very properly within our model. So besides the transporters, of course, we also wanted to see if our neurons actually showed some functionality. So we did a calcium assay, and you can see here in the pictures that indeed our neurons did show spontaneous electrophysiological activity, shown with the lit up regions right here, and also by the electric stimuli that are measured here. So coming back to the barrier, which is quite crucial for the blood-brain barrier and is very necessary to study compound-induced barrier disruption, we use a piece of equipment called the organoteer, and the organoteer fits nicely on top of our organoplates, and it can measure the transendothelial and epithelial resistance of all the 40 models in under 1 minute. So that really helped us with compared to, for example, the chopstick-like method, where you have to go well by well. And so how does an assay like this work? again, we grew our barrier tissue, so in this case, the human-derived brain endothelial cells in that top channel, and then we add a fluorescent dye to see if we indeed can have a leak-tight vessel. So this is like a BI assay where a leak-tight vessel will show no overspill of the fluorescent signal into the adjacent channels, whilst a vessel that is leaky due to, for example, compound disruption, shows leakage of that fluorescent signal into the adjacent channels. So we tested that with a barrier integrity assay as well as with the organoteer. And in order to do so, we used a component called staurosporin, which is known to cause disruption of barrier and kill cells. So what we can see here indeed is that the staurosporin disrupted our barrier. We see that both by leakage of the fluorescent signal into the adjacent channels, both in the pictures here as in the graph, and actually the dose-dependent effect, so with a higher concentration, we actually saw a fluorescent signal that is coming close to a cell-free control that we performed. I'd also like to point out that for the timelines, we directly compared barrier integrity assay with our tear measurements, and what we saw is that the tear measurements were able to pick up those disruptions a little bit earlier than the barrier integrity assay does. So that's very nice to have that confirmation of the system. Now going to stroke, so now we have a functional neurovascular unit, and we want to model what happens in the stroke. So there's 3 components that we use to model this disease. No glucose. So we used a glucose-free medium for that. No oxygen, because of course if the clot disrupts the blood flow to the brain, there's also no oxygen flowing there, and we modeled that by using antimycin A, which is a complex 3 inhibitor of the electron transport chain. And then we also halted perfusion by simply stopping the rocking from happening for 16 hours. So in this way, we modeled our stroke by a 3-fold approach. And I'd like to point out here as well, just like Donald Goubert was saying in his nice keynote lecture on Tuesday, how important exactly those dynamic conditions are as well. So what we did here is we looked at a combination of both the presence or absence of glucose and oxygen together with perfusion as well. And what we could see here, we are looking at the perfusion compared to the HB-MEC free control, and what we could see is that when perfusion is actually halted, the impact is much higher than when the perfusion is present, really modeling here the ischemic stroke conditions in which there's also no perfusion towards the brain presence anymore. Then another important aspect of ischemic stroke is the reduced mitochondrial potential. And again, we compared the different conditions with each other, seeing that the static conditions showed the biggest response compared to the perfused conditions, also with regard to the reduced mitochondrial potential. Then another important aspect of modeling ischemic stroke is the reduction of ATP. And we looked at this actually both at the blood side of our model as well as on the brain side. So what we could see is that the ATP concentration after halting the perfusion as well as taking away that oxygen and the glucose really leads to a reduced ATP concentration both in the blood side as well as on the brain side. So you can really see that the conditions in which all 3 of the stroke properties are present have the biggest impact on all 3 of the aspects that we see in stroke as well. So to summarize, our 3D in vitro model of the neurovascular unit has a triculture consisting both of the human-derived brain endothelial cells, the neurons and the astrocyte. We were able to show that it displays relevant markers both for the transporters as well as for the activity. And the barrier is formed in the model, which is leak tight. We tested this both with organotear measurements as well as with a barrier integrity assay. And I also like to point out here that values don't always say everything. So even though we might always be looking for super high tear values, for example, we actually saw that in our human-derived brain endothelial cells, tear values were not super high, whilst the small molecule fluorescent dyes were contained, so showing that it is indeed forming that leak-tight barrier. Now, once we had this model, we were able to also model the disease, so stroke, by using a threefold approach, inducing the chemical hypoxia, hypoglycemia, and by simply halting the perfusion, really mimicking the conditions that also happen when a blood clot disrupts the flow to the brain. And then the resulting cultures that I showed to you today showed a reduced barrier integrity, a reduced mitochondrial membrane potential, as well as reduced ATP levels. So we really believe that with this model, we are able to give a good representation of what actually happens physiologically after stroke, and to then use this model to test for better therapies. So like we are all here, also we at Mimetas really are in the field to unlock novel biology, to create better and faster assays, and to eventually have an impact on therapy development and patient lives. So with that, I would like to acknowledge all my colleagues from the Netherlands, as well as from Japan, as well as from the US, and all of our collaborators also in the Entrain project. So with that, I'd like to thank you for your attention, and I'd be happy to answer any questions.